So this is just kind of a shorter video discussing some of the interesting stuff in relation to GLP-1 acting as a neuromodulator on these neurons called POMC or CART neurons. And there's these neurons within the arcuate nucleus of the hypothalamus. They go out and they project to, or they innervate neurons within the periventricular nucleus of the hypothalamus. And generally these neurons, these POMC neurons, or these pro-opiomelanocortin releasing neurons, tend to be pro-satiety. So they're inducing a state of satiety where you're no longer hungry. And they're kind of the opposite of appetitive. So they almost inhibit like an increase in the appetite. Whereas there's these other neurons that are functionally antagonistic to the POMC neurons called the AGRP neurons or the agouti neurons, or they also release neuropeptide Y or NPY, and they're functionally named based on what they release. So these guys, the agouti related peptide neurons and the NPY neurons, they're releasing those two neurotransmitters, and they're pro-food intake. So they're appetitive, they're promoting the organism to go out and seek food, whereas these ones are causing the organism to decrease food intake, but also they relay information to the nucleus tractus solitarius and also the sympathetic nervous system. So they're influencing the vasomotor center to functionally decrease food intake, but also they're increasing energy expenditure. So they're saying, okay, I'm full. Now's the time not to be eating, but to expend energy so I can keep myself at a homeostatic balance in terms of my energy levels and the amount of resources that I'm currently storing. And so these POMC neurons are influenced by neurons that are within the nucleus of the solitary tract. And so there's these neurons called pre-proglucagon neurons, and they go over and their cell bodies are within the nucleus of the solitary tract, and they go out and they reach their axon out and influence these POMC neurons. And what they do is they release GLP-1, not as a neurotransmitter, but as a neuromodulator. And that GLP-1 has an effect on GLP-1 receptors, which are stimulatory G proteins. So this is my little G protein right here. This is the receptor. These are the little transmembrane domains that kind of go in and out of the membrane. But GLP-1 can bind to that receptor. And this G protein is part of the secretin class of G proteins, but it's stimulatory. So if GLP-1 binds to this guy, it changes the shape of the G protein linked receptor or the coupled receptor, which will activate a G protein. And by activate, what that means is usually GDP is bound, and GDP is kicked out, and GTP comes in and binds, and that allows this little G protein to dissociate from the receptor and float over. And what it does is it binds to another transmembrane protein called adenylate cyclase, which what that does is it converts ATP into cyclic AMP, CAMP, so cyclic AMP, which is functionally a second signal. So GLP-1 would be considered the first signal right here. The second signal molecule in this signaling cascade will be cyclic AMP. And cyclic AMP has an interesting effect because it can activate kinases within the cell that can go and phosphorylate proteins, but it can also go and bind to this little channel right here. And this right here is what's called a funny channel. It's also known as an HCN channel, but it's funny because most channels that are voltage gated open when you have more of a positive membrane potential, when it's depolarized. So typically channels will open when you start getting little positive charges in this local area. But this funny channel is funny because it'll open when the channel is more negative. So when you're at resting membrane potential around -70 or -90, what happens is this funny channel opens. So the channel opens and it allows sodium to move from the outside of the cell to the inside of the cell. And that sodium will depolarize the membrane. It'll cause these L-type calcium channels to open, and that'll cause a wave of depolarization down the axon, causing an action potential. And then there's these other channels called voltage-gated potassium channels, which I did not draw. But what happens is potassium leaves the cell through those voltage-gated potassium channels, which will functionally reset the neuron back to its resting membrane potential at around -70 to -90. And it may be a little different in these POMC neurons. I'm not entirely sure. But if I reset the neuron back to around -70 or -90, this channel, this funny channel, will open again, allow sodium to move in, and it'll depolarize the neuron again and send another action potential. So these cells, the POMC cells, are tonically active. So they're always firing action potentials. They're sending one, it resets, and then the funny channel opens again, depolarizes, then they send another. So they're always firing, and it's kind of a rhythmic activity. But what happens is this GLP-1, the presence of GLP-1 as a neuromodulator, can influence the rate that they fire. So they're tonically on, they're always firing, but I can crank up the rate that they fire where they're firing more often. And if they're firing more often, functionally, those POMC neurons are decreasing food intake and increasing energy expenditure. So the more they fire, the more they have that effect. And it's through this molecule, cyclic AMP, because it goes over and it binds directly with this funny channel, causing it to open with less repolarization. So you don't have to get the membrane quite as negative in order to open this channel and allow sodium in. So it's causing the cell to fire more often. Thanks.